Okay, I think we can start. Well, my point is that if there's an echo and we have some problems to understand each other accents, then we have a double problem here. Okay, so, okay, we'll start and see how it goes, okay? Okay, so good afternoon, everyone. Thanks for coming. It's Friday, close to the weekend. And so today, after the introduction we had yesterday, we will start getting deep into some fundamental concepts on synthetic biology. And as I mentioned yesterday, the whole story about synthetic biology is based on the so-called gene expression flow. That is the process through which different biological systems, and as I anticipated yesterday, in particular bacteria, that is the main matter of this course, convert the information that is in DNA all the way to a phenotype or a property or an activity that we can measure, monitor, and observe. And well, let's get started. So what you have in front of you is the so-called central dogma of molecular biology. I have to admit that the term dogma I don't like, because dogma means that this is like this, period, it will always be like this, and it will never be changed. So as you will see right now and also later in the course, this is not completely correct, because there are some details that do not fit completely with this central dogma. But in general, biology and molecular biology takes for granted, and it's a good assumption, that the flow of information between DNA, RNA, and proteins go unidirectionally. So this was proposed a long time ago by the same guy that discovered the structure of DNA, and basically has remained like that for a long time. So there are some qualifications that we have to make to this central dogma, because we know that DNA copies itself, so it replicates, so you have this loop there, and also it is possible, and it was discovered time ago, that RNA can also be retrotranscribed into DNA, and therefore, at least in the case of RNA, you can go up from RNA to DNA in the different direction and the central dogma. And also, you know that some RNA viruses are able to self-replicate, ignoring completely that you have DNA. So this is a real qualification. But then after you have RNA, then you go into proteins. And so far, it has been not possible to go from proteins back to RNA or to DNA. So in this upper part of the central dogma, you may have some, say, information going back and forth, but once you get into RNA and RNA get translated into proteins, then there's no way down or way up. So, but we can keep on qualifying the central dogma, because at the same time that the DNA eventually encodes proteins, it also encodes metabolism, because metabolism is mediated by these proteins. So let's take for granted now that this is true, and we are going to operate in most of our course on the basis that we have an information flow going from DNA to metabolism. Well, in fact, this reflects something that what you may call the genocentric syndrome, in which everything is DNA. DNA, 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 and more DNA. And everything is subject to the orders given by DNA. So it's like the DNA is the master of the biological show, and all the other actors are just actors of all the instructions and all the power that DNA has. So also, as you will see later in the course, this has to be qualified as well. But for the time being, let's consider that everything that we need to program or to modify the behavior, the properties of biological systems, eventually have to go down to focusing on DNA. So the extreme view of that is the idea that biology is about selfish DNA, selfish gene, and the entire, say, agenda of biological systems is self-perpetuate their DNA. Again, this is something that we could discuss at some point. It's a kind of a strong statement, and perhaps it's not completely true, but again, most contemporary biology and most contemporary molecular biology live out of this dogma, this selfish DNA, and this entire focus on DNA as the ultimate source of biological activities. All right, well, I hope that we don't run often in this technical process. Well, anyway, so what we're going to do today is to examine in some detail, and there are some parts of my presentation that will be a bit tough to follow, so I'm sorry, but I have to deliver that contents. And also, as you will see, we have the standard gene expression flow, but through synthetic biology methods, we can make like diversions from the standard, say, flow, and then create new properties that are not yet or have never been evolved in nature. Okay, just to recap, this is what I showed you yesterday. You have DNA, you have RNA, and you have ribosomes and different stages. And as I mentioned yesterday, in every step of the way between DNA and proteins, there are many, many checkpoints that can be subject to additional regulation. So what I'm going to do is to run through various major or important steps in the whole process of transcription and translation. Some of you might be familiar with them. Others yesterday told me that they were not so familiar, so please be patient and understand that I have to go through this just to make sure that everyone knows what we're talking about. Okay, so you have a gene in the genome, in the DNA, and then first the gene is transcribed, and then once it is transcribed, then you generate an RNA, and then this RNA is immediately recognized by ribosomes, and as a consequence of the recognition and the binding, then proteins start to pop out from the ribosomal body. So this would be in a kind of a snapshot what happens during the whole gene expression flow. Okay, now what happens is that transcription itself and translation itself is a multi-step process. And let's go with some detail on what is going on here. Well, first, what happens for transcription to occur is that RNA polymerase has to bind a specific sequence of DNA that is called a promoter. I will give you more details on what a promoter is. And then when the RNA polymerase binds to that particular sequence, then the two strands of DNA separate. And then one of the strands start being recognized by the RNA polymerase, and because of the enzymatic activity as a polymerase of the RNA polymerase, then an mRNA starts being synthesized. One thing that is important, and I will return again to that later, is that the recognition step of the RNA polymerase is mediated by something, by a protein, by a component of the RNA polymerase complex that is called the sigma factor. So the sigma factor is the part of the RNA polymerase that recognizes specifically a given promoter sequence. So there are various sigma factors, and depending on the sigma factor that is associated to the rest of the polymerase, then different promoters can start transcription or not. So the moment transcription starts, then the sigma factor is left behind, and then the rest of the polymerase continues its, say, way through the RNA polymerase until it finds another sequence that is called a terminator. So in fact, you know that it's intuitive. You have a promoter where things start, and then you have a terminator that is where things terminate. So what happens then? When the polymerase hits a terminator, then what happens is that the polymerase stops, and then the mRNA is released, and then the RNA polymerase is released as well. And it becomes available for another round of transcription. So the main components of this first step in the transcription process that is part of the gene expression flow includes the RNA polymerase, that is the basic enzyme that runs the whole show, the sigma factor that enables a specific recognition for a given DNA sequence, the raw protein that in some cases mediates termination, in other cases it does not, the promoter and the terminator. So each of these, actors here can become parts, as we defined yesterday, for synthetic biology. We can have parts that are sigma factors, parts that are different types of RNA polymerase, parts that are promoters. There are many, many parts that are different promoters, and parts that are terminators, okay? So all these actors appear naturally, but through synthetic biology, we can make combinations that are not natural, and therefore we can create new capabilities. Now, the most important of every biological part is the promoter. What is a promoter? Okay, in a very simple definition, a promoter is a binding site for an RNA polymerase. There are different types of RNA polymerases. So there is a default RNA polymerase, and then there are specialized RNA polymerases. So default RNA polymerase is composed by various subunits that you will see in a moment, but for the most part, in the genomes of prokaryotes, it recognizes specific sequences that are defined by two motifs. One of them is what is called the -10 region, and then the -35 region. One thing that I ask you is that I'm going to mention and define many terms, many words that are important for the rest of the course, because we will be mentioning them very, very frequently. So the RNA polymerase will recognize and will bind a DNA sequence when, at a given specific distance, you have this -10 region and this -35 region that, the consensus is that you have TA, TTA, and in -35, TTG, AC, whatever. Okay. Then a few nucleotides later, if this interaction is productive, then transcription starts. And starts at site what is called +1. So the first nucleotide that is converted into an mRNA is the +1, say, transcript, or the +1 position of the transcript, and then transcription continues. So the region of DNA that is before the binding of the polymerase is what is called an upstream region. And what happens later is the downstream region. Okay. Important terms: transcription start sites, -35 region, -10 region, upstream, and downstream. Terminology that, please, you have to get familiar with. Now, more jargon, more terminology to define transcription and to understand and to describe transcription. You have a transcription start point, that is the +1 location that I told you before, and then you have DNA that is more proximal to this start point and more distal to this start point. Again, new words: proximal sequences and distal sequences. The distal sequences can be distal downstream or distal upstream. And you will see that there are promoters where these regions are important depending on where they are downstream or upstream that you have them there. Okay. Now, more terminology. When the RNA polymerase binds the polymerase, the RNA polymerase binds the target DNA, the promoter, then what is formed is called a closed complex. It's just a recognition, nothing else. But then, if the interaction is productive, then the RNA polymerase go on, and then not only binds the DNA, but it opens the DNA strands at the site where the polymerase binds, and then is formed what is called an open complex. The difference, or the main difference between this one and this one, is that in a closed complex, it's reversible. You can stick the RNA polymerase to the promoter. But once an open complex is formed, then the polymerase is committed, and then it cannot go back. It really gets stuck there, and it has no other possibility but continue transcription. And then when that happens, you go into elongation, and it's where the RNA polymerase gets rid of the sigma factor and starts reading the rest of the DNA to generate an RNA. Okay? Again, important words: closed complex, open complex, commitment, and elongation. So these are all words that, please, you have to remember for future, say, discussions. Now, the polymerase moves, and then the RNA chain grows longer. It's like something that you read, like a machine that goes on, and then you read the DNA and convert that into RNA. Is why it's called transcription. Okay, then it continues and continues and continues, and the moment that the RNA polymerase proceeds, then the region where the polymerase goes is unwound, is the two strands of DNA are separated to allow the polymerase to read the sequence, because otherwise it would be difficult to read the sequence. And then once the polymerase ends or meets the end of a gene because of a termination, of a terminator, then you have this phenomenon called termination. So the terminator makes the polymerase to drop off the DNA, and then you have terminator. So the transcription of the genes stops altogether. Okay, so this is a very simple cartoon on how this works. So this is the DNA here. Then the RNA polymerase gets stuck to it. It looks, I mean, in this cartoon it looks like a little insect. But what it is that the RNA polymerase, or the default, the housekeeping RNA polymerase in bacteria, is a multi-protein complex where you have various units. And this is symbolized here with different colors and everything. So this red ball would be the sigma factor. That is the one that recognizes the target sequence. And then what happens is that the polymerase binding makes the DNA to wrap around the rest of the polymerase, and this kind of kinking what makes the two strands of the DNA to separate. So, and then there's a separation of strands, and then the polymerase continues its route. So the RNA polymerase, as I said, is a multi-protein complex formed of two big subunits, the beta and beta prime subunit, that is associated to what is called an alpha subunit. So there are beta and beta prime, and there's a dimer of the alpha subunit. So this is the whole, say, structure, and then you have this sigma subunit that is associated to the other two. So that means that the default, the housekeeping RNA polymerase of prokaryotes consists of a multi-protein complex. It's not just one protein, it's a multi-protein complex. However, as we will see later, there are alternative RNA polymerases that consist of a single unit. For instance, the famous T7 phase polymerase, where everything is in a single protein. And this polymerase, the 47, is very useful in synthetic biology, as you will see a bit later. Well, these are just to show you that these cartoons that I show you in a very, very simple fashion are now known. I mean, the structure of the RNA polymerase is now known to a tremendous detail, and we know where the DNA goes, how it binds, where the RNA emerges and everything. So just to mention that there is an extraordinary mechanistic detail on how this works. So what happens inside the polymerase? Okay, so many things happen. This is an amazing molecular machine. And what happens is that as the polymerase goes through DNA, you have like various occurrences at the same time. So there's locations where the DNA is rebound, say, and then the RNA polymerase recognizes the coding strand. Then you have a window of sequences where the two strands are separated. You have a catalytic site, that is the moment where RNA is produced, and you have to be aware that only one of the two strands is re-recognized. The other just is there to complement and to maintain DNA structure. Now, engineering. Biologists will describe, we have a promoter, the polymerase binds here, then the polymerase proceeds, the DNA separates, and so on. But let's put some numbers behind that. So in every promoter, you have a number of parameters that you have to consider for any serious synthetic biology. And all of them are summarized in this slide here. So in a promoter, you have what is called a basal level. That is the transcriptional activity that you have by default when the promoter is just active without any further action of any additional proteins or activators or anything. Then if the promoter is inducible, and this is the case in many promoters, then you have the possibility, you have a parameter, is the induction rate, and the induction rate is connected also to inducibility. That is the rate of induction that you have after a given time until you reach a steady state. So depending on the conditions, the steady state can reach a given level, but in other cases you can have a maximum capacity for transcriptional output. And this is something that you will see some examples that happens very frequently. So in other words, you have a promoter that has a given activity, and even in the best possible scenario, it reaches a limit. So it can go beyond a given limit. So this is called capacity, and this would be in this case the maximum capacity. Then the induction is the ratio between the basal level and the induced level, and then the inducibility is how it goes a long time. Okay, induction rate. So this is something that in many promoters, mostly those which are inducible by an effector or by some physical signal, these are parameters that are important to determine in a given specific project what type of promoter you use or what type of behavior you want to have in your construct, because not all promoters are the same. In fact, every promoter has a kind of personality, and depending on the specific project that you are interested, then you may have more or less of these parameters here. Okay, so promoters, by default, they could run only with RNA polymerase. So you have a good binding sequence for RNA polymerase, and you have the polymerase that sits on this recognizing sequence, and then you have transcription. That happens, but it's not a good idea for bacteria to have promoters just like that. So promoters, in most cases, are regulated. So regulated, it means that the transcriptional output that you have at any time depends on the environmental conditions. And that makes a lot of sense because as bacteria and other living systems are exposed to very variable environments, they have to adapt their gene expression levels to the environment that they have at any time. And therefore, it makes sense that promoters have evolved to be regulated on the basis of environmental signals, of environmental cues. Again, we have a similarity with engineering. So engineers, one of the big, contributions of modern engineering is the ability to control fluxes and to control processes in an automated way. And this is just one of the many devices that engineers have developed over time, for instance, to control a flux of a liquid, to make it maintain a given level, regardless of what is the strength of the flux that comes in this way. So you have, this is rotating, and depending on the flux that you have here, then the balls will go with a wider angle or a smaller angle in such a way that the flux will be maintained constant. So interestingly, this type of, scheme that engineers found or developed in the 19th century, something similar conceptually can be found in bacteria and in many similar in other biological systems. So why do you want to have transcriptional regulation? Well, basically for three reasons. One is that transcriptional regulation enables or allows you to detect and respond to nutrient availability. So for instance, you have genes for degradation of glucose. It makes no sense that you express this gene in the absence of glucose. You just express these genes when you need them, not before. And by the same token, with many other nutrient availability circumstances. The other is processing environmental signals to make decisions. For instance, if you have a toxic compound in front of you, then you have to make a decision of whether you are going to eat it or you are going to run away, for instance. And because of the transcriptional regulation that many promoters have, bacteria can make these decisions. And they can also process various signals, because it's not that one nutrient is toxic, entirely toxic. It can be a bit toxic. So depending on the concentration, depending on the temperature, depending on the surroundings, then bacteria can make the decision of degrading it or not, or running away, or different other behaviors. And also, and this is very important, transcriptional regulation and regulation in general enables bacteria to adapt to physicochemical stresses. Physicochemical stresses mean heat, cold, solvent exposure, desiccation. So these are environmental signals that bacteria recognize, and they convert these signals into inputs in the promoters through different types of mechanisms, as we discuss in a moment. Another super important feature of transcriptional regulation that is important for synthetic biology is that regulation can happen through two mechanisms, through a mechanism of negative regulation and a mechanism of positive regulation. So in the case of positive regulation, what happens is that in the case of negative regulation, that is the simplest way of regulating something, what happens is that the polymerase cannot access the promoter because the access to the sequences of interest are blocked by a repressor. So this R protein here is a repressor, and you have a promoter here with a polymerase, and the polymerase would like to go in this direction, but if you have a repressor that blocks the pathway of the polymerase for transcribing DNA, then you have no transcription. Okay, so this would be the easiest, the easiest way to regulate something. You have a flow of water in your house, and you want to stop it, all you have to do is to block it, and then it will stop. So by the same token, regulators, repressors will exert their regulation on the basis of blocking RNA polymerase, say, way to transcribe DNA. Another more complicated way of regulation is positive regulation. So positive regulation involves that the RNA polymerase has some affinity for the promoter, but the affinity is not great. So it needs the help of another factor, the activator, to recruit the RNA polymerase to the promoter. Okay? And therefore, you have an active recruitment of the RNA polymerase to the place, and then you enable transcription. So just please keep in mind, because again, it's very important for the type of stories that I will develop later. So positive and negative regulation, both of them work, but the mechanism is different in each case. So as I said, repression is the simplest regulatory mechanism, because all you need is to have a protein that sits in the way of the polymerase in such a way that it will block the preceding, proceeding transcription. The site for binding the repressor to the DNA to block transcription is called an operator. So you have the promoter, then you have the operator that overlaps in some cases with the promoter itself, and in other cases is just adjacent to the promoter sequence, and then the polymerase will bind to the promoter, but the regulator, the repressor in this case, will bind the operator. And also, depending on the type of repressor, then the repressor may bind or may not bind depending on whether you have an effector, something that you add from the outside. And then you generate many kind of, states. So you have a repressor that binds by itself, but unbinds when it is exposed to an inducer, for instance, the super famous LAC-Y repressor. Then you have a type of regulation, but there are other repressors that normally do not bind the DNA, but do bind in the moment that they see an effector or an environmental signal. So you have all these possibilities. So look at the entire panel of possibilities that nature has invented to do this, that obviously will be later leveraged by synthetic biologists to make the circuits and the type of things that we discussed yesterday. So, well, this is just a follow-up of the previous slide. So the repressor will block the kind of course of the polymerase, and the polymerase either cannot bind or will simply be blocked in its way to transcription of the rest of the DNA. Okay? Okay, so the sites recognized by repressors in many cases can be identified easily just by looking at the DNA sequence, because practically 99% of all known repressors bind to inverted repeats, what is called palindromic sequences. So palindromic sequences means sequences that can be read identically in one direction and the other. And, well, it has been known for many years now that most repressors have motifs in the protein structure that are evolved precisely to recognize these, say, inverted repeats in the DNA. So sometimes it's very easy, and a computer can do that, to screen a large segment of DNA for inverted repeats. It's an indication that a repressor is binding there. And it is known that there are these motifs that is called helix, alpha helix, that are present in many, many repressors that recognize cognate inverted repeats in the DNA. So that's something that is kind of known. Repressors. But now activation is more complicated because, as I said, it's not just about blocking the way of the polymerase into transcription, it's about recruiting the RNA polymerase to the promoter in such a way it can initiate transcription. So in that case, instead of having the binding site of this regulatory protein downstream or overlapping the RNA polymerase, in this case, systematically, the activator has to bind upstream of the binding site of the polymerase here. So that once the transcriptional activator binds to this site, it acts as a magnet that will make the RNA polymerase to dock there and then initiate transcription. In many cases, this transcriptional well, not in many cases, I would say in 100% of the cases, transcriptional activators are themselves regulated by an input signal. So that means that the transcriptional regulator by itself will not bind or will not do much to the system in terms of binding DNA, but when there's a signal in the environment, whether a chemical signal or a physical signal, then it will bind the DNA and it will act, as I said, as a magnet to bring the RNA polymerase to the right position in the promoter to initiate transcription there. Now, sigma factors. I mentioned them on passing, and just to say something about them. So sigma factors are the proteins that belong to the RNA polymerase complex that endow specificity of the whole polymerase for a given sequence. So in bacteria, most sigma factors, the immense majority of sigma factors, belong to the so-called sigma 70 family that recognizes this -10, -35 sequence that I was talking about before. So, well, there's a bunch of them with different names, with different genes and so on. And the trick in its case is that depending on what type of sigma factor associates to the rest of the polymerase, then the sequences in the DNA that are recognized by the whole enzyme, what is called the holo enzyme, will vary depending of the circumstances. There is a special type of sigma factors that is a different family. It's called the sigma 54 family. And this is a class in its own. It's very different from all other sigma factors. And it's different because in this case, the activation of the corresponding promoter is not related to the -10, -35 sequences, but to another motif that is called, or are called, the -12, -24 sequences here. Okay? And the trick here is that the polymerase is always sitting, instead of having, of the other cases with the sigma 70, where the polymerase sits in the recognized sequence and it proceeds immediately. In this case, the polymerase that is associated to sigma 54 sits in its target site, but it stays there. It does not move. And to proceed with the transcription, it requires an activator that binds not close to the polymerase, as was the case with other activators, but far away from the polymerase. So this is interesting because you have the polymerase binding site here, you have the activator binding far away, and obviously the only way that activation can happen is if you have DNA bending in such a way that the transcriptional factor that is bound farther upstream
from the polymerase can have a contact with the RNA polymerase here. Okay, so one typical, well, one say known member of this family of transcriptional regulators is called anti-RC. There are many others, and as is sketched here, to have activation of the downstream polymerase with this upstream anti-RC, then you need to kind of bend DNA in and form a loop here. So this is something that again has been leveraged in some cases for making funny constructs. Okay, one more, cork screw. In many cases, promoters, even promoters that are completely natural, regulated both positively and negatively, okay? This is something that, one more time, synthetic biologists leverage for their own purposes, but this is something that occurs in many prokaryotic promoters, in many bacterial promoters. And the super typical example is the lag promoter of E. coli. That is perhaps the most studied promoter of the whole history. So in this case, we have a regulator, a negative regulator, that is the lag repressor, but at the same time, you have a positive regulator that is called the CRP protein. So by having a promoter that can be subject to both positive action and negative action, you can have the promoter acquiring a large number of states depending on the circumstances where the promoter is. So, for instance, if you have glucose only, then the lag repressor is the one that represses the thing. You have lactose only, then what happens is that the activator is fully active, and then it will make the RNA polymerase to proceed. You have none of them, then the system will be off, and you have both of them, the system also will be off. So just by having in the same, say, control unit the possibility of entering a repressor and an activator, then you can have in the very same promoter up to 4 different states depending on the inputs that you have at the moment. Okay, well, this has been a bit kind of tough and heavy and everything, but you know, this is part of our duties to explain. But there are ways to express that in a more, say, funny way. So one of or some of the contents that I just told you can be read in a completely different style, in a completely different narrative, in this paper that I will share with you later, wrote by Drew Endy, that is one of the big guys in synthetic biology, in which he explains and elaborates about all these tricks in transcriptional regulation and how they can be applied and leveraged for synthetic biology and so on. By the way, I want to give you an advance, and is that before we finish today's classes, I will make an assignment of some papers for you to read and for you to present sometime next week. So you have something to do over the weekend. So we'll discuss that later, in any case. Okay, so so far this has been like the toughest part of today's classes. The rest of the class will be like lighter and more interesting. I think that it's important that you mostly understand the nomenclature: upstream, downstream, terminator, activator, repressor, inducer. I mean, all these are keywords that we need to understand and to comprehend well and to really proceed with our agenda for today. Any question? Okay, yes. Factors are associated to specific environmental conditions. For instance, there's a sigma factor activated with heat, with high temperature. Why? Because high temperature environmental condition that is extremely dangerous, and then what happens is that this sigma will be expressed and will take over all the entire transcriptional machinery to activate only and tolerance to heat. because, you know, are you don't care. The same token, the same, say, logic, bacteria have in such a way that the moment that they find a over-imposing environmental condition, this, say, play of different transcription factors will ensure that at a given point, all transcription to just the number of genes, everything business as usual. Okay, so in fact, I think that that's, well, you know, many, well, I would say most people, most biologists, most neurons or you name it, think that bacteria are very simple. You know, you just have a sack of enzymes there, DNA, it's very, very simple. No. Bacteria are super, super sophisticated, and keep in mind that bacteria were on Earth much before we arrive here, and bacteria will remain on Earth much later that we just become extinguished. So bacteria have elaborated, super sophisticated ways of dealing with environmental conditions. Study them in detail. Amazing. And basically nearly every single mechanism that you can imagine that we use for, you know, dealing with our daily life, the mechanism has been invented before by bacteria. Maybe not with the same, say, implementation, but with the same logic frame, with the same relational logic. And that's something that to me is quite amazing. So when bacteria are subject to a given stress, in some cases, there is one sigma factor that takes over, displaces all the other sigma factors, and makes sure that every molecule polymerase in the cell just makes what survive that particular condition and not the other, okay? So sometimes, you know, bacteria have to decide many things, you know, so you have a nutrient here, you have a nutrient there, you have a nutrient there, and then under normal conditions, they can, you know, when you have the coffee there, the samosas, you have tea and so on, and you can go, well, I prefer to have a tea or prefer to have a samosa or to have, but then if there's a fire alarm or you want to do is to run about everything. So the same happens with bacteria. So the moment that they are stressed, they forget about everything and just concentrate in surviving the stress. So that's an interesting point of view. So I know that some of you here are interested in neurobiology and so at some point we can discuss whether neurons are smarter than bacteria or what, right? So we'll discuss that at some point. Well, anyway, so it's 5 minutes to 3. I would propose to make a real pause of 5 minutes because yesterday I think we had a super long first session. So what I would do today is to, you know, not to get to the hour, but to leave you for 5 minutes break before we get into the second hour. So you can go to the bathroom or have a drink or something. But please, let's start again at 3 sharp, okay? So have 5 minutes break now, and then we'll continue in 5 minutes. Great.